Your immune system has an innate immune response, which is immediate and nonspecific, as well as an adaptive immune response, which is delayed and specific. The adaptive immune response is carried out by lots of unique B cells and T cells, which are highly specific for pathogens based on their unique parts, called antigens. Now, focusing on just T cells, they can only bind those antigens, which are typically short peptides, when they're displayed on a major histocompatibility complex, or MHC molecule, which is sort of like a silver platter that's on the surface of a cell. The MHC molecules are also called human leukocyte antigens, and these proteins are encoded for by MHC genes, which are found on chromosome 6. There are actually two groups of genes. One group of genes encode the MHC class I molecule, which is bound by the CD8 molecule on the surface of cytotoxic T cells. Another group of genes encode the MHC class II molecules, which is bound by the CD4 molecule on the surface of helper T cells. MHC class I genes encode the proteins HLA-A, HLA-B, and HLA-C, which is easy to remember, since for MHC1 it's always one letter. MHC class II genes encode the proteins HLA-DP, HLA-DQ, and HLA-DR, which is also easy to remember because for MHC class II there are always two letters. And these genes are called histocompatibility because they're really important in determining whether or not a transplant is compatible or gets rejected. But their role isn't just to wreak havoc on transplants. They're critically important in making sure T cells recognize and react to antigens. And even though they're called human leukocyte antigens, they're not just found in leukocytes, or white blood cells. HLA proteins that code for MHC class I molecules are found on all nucleated cells throughout the body. Even platelets, which are fragments of nucleated cells. In fact, the only cell that don't have them are mature erythrocytes, which don't have a nucleus. MHC class II molecules are only expressed on antigen-presenting cells, like monocytes, macrophages, dendritic cells, and B cells. The MHC class I molecule has two protein chains. A larger alpha chain, which contains both a peptide binding groove and a transmembrane region which anchors the MHC class I molecule onto the cell surface. And there's also a beta-2 microglobulin chain, which is linked to the alpha chain. The alpha chain has three extracellular domains: alpha 1, alpha 2, and alpha 3. Alpha 1 and alpha 2 make up the peptide binding groove. The beta-2 microglobulin basically props the whole structure up by binding between the alpha 1 and alpha 2 domains and the alpha 3 domain. The peptide binding groove binds peptides that are approximately 8 to 10 amino acids long, with many hydrophobic residues that'll bind easily to the hydrophilic amino acids inside the peptide binding groove of the MHC molecule. MHC class I molecules allow immune cells to sample proteins from within your cells. To do this, MHC class I molecules use what's called the endogenous pathway of antigen presentation. When a cellular protein is marked for degradation, it's sent to an organelle called the proteasome, which is found in all nucleated cells. The proteasome degrades the protein into short peptide chains, which are then transported to the endoplasmic reticulum using proteins called transporters of antigenic peptides, or TAP for short. Meanwhile, the MHC class I molecule is being made with both the alpha chain and the beta-2 microglobulin, and this is also happening in the endoplasmic reticulum. The TAP proteins load the short peptide chains onto the peptide groove of the MHC class I molecule using a molecule called tapesin. The MHC class I molecule and peptide then goes from the endoplasmic reticulum through the Golgi apparatus and into an exocytic vesicle bound for the cell surface. Circulating cytotoxic T cells as well as natural killer cells, which are innate immune cells that can directly kill target cells, can then interact with the MHC class I molecule and the antigen. If no recognition happens, then the cell lives. But if these immune cells do recognize the peptide that's being presented as foreign, then the cell gets attacked. The whole system is a bit like taking the trash and putting it on the corner each week. This allows the immune cells to investigate and know what's going on within the cell on a regular basis, and can lead to early identification of a viral infection, or abnormal protein synthesis in a cancerous cell. If MHC class I molecules are designed to present antigens from inside the cell, then MHC class II molecules are designed to present antigens from outside the cell. These are cells that engulf and destroy pathogens and then present antigens to CD4 positive T helper cells. The MHC class II molecule has two chains, an alpha chain and a beta chain, and each chain has two domains. Both chains penetrate the cell membrane and contribute to the binding of the antigen at their alpha 1 and beta 1 domains. The MHC class II molecule has a groove that can bind much larger peptides, 14 to 20 amino acids long on average. MHC class II molecules use the exogenous pathway of antigen presentation. The exogenous pathway starts when a pathogen or a protein is ingested by an antigen-presenting cell and brought into a vacuole called the endosome. Granules within the phagocyte fuse with the endosome, forming a phagosome, or phagolysosome. These granules are filled with proteolytic and degrading enzymes which degrade the pathogen or the protein into many small peptides. Meanwhile, the MHC class II molecule is synthesized in the endoplasmic reticulum, with all the other host proteins, including MHC class I. Instead of acquiring its peptide from within the endoplasmic reticulum, like an MHC class I molecule, the binding cleft or groove of the MHC class II molecule is filled temporarily with a molecule called the invariant chain. The MHC class II molecule and invariant chain is then transported through the cell in a vesicle where the invariant chain is degraded. The vesicle with the MHC class II molecule then fuses with the phagolysosome, and the peptide cleft is filled by one of the peptides in the phagolysosome. This vesicle now contains the MHC class II molecule that's bound to the peptide antigen, and it's transported to the cell surface where it can present to circulating helper T cells, which can go on to stimulate the immune system. Alright, as a quick recap, the MHC molecules or HLA proteins are encoded by MHC genes. MHC class I molecules include HLA-A, HLA-B, or HLA-C, and they present antigens from within the cell using the endogenous pathway and are presented on all nucleated cells throughout the body. MHC class II molecules include HLA-DP, HLA-DQ, and HLA-DR, and they present exogenous antigens from extracellular pathogens using the exogenous pathway, and are found on professional antigen-presenting cells,